Good morning, children. This is Mrs. Anjum Aleem from Springfields School, Masabtank, biology teacher for class 10. I am going to continue with the lesson number 6, reproduction. In my last video, you have watched different modes of asexual reproduction. Today, we shall be talking about the vegetative propagation. What is vegetative propagation? Again here same, vegetative propagation means mostly in higher plants. It takes place in higher plants and it may be natural propagation, vegetative propagation, or artificial vegetative propagation. So again, vegetative means it's another type of asexual reproduction only. It does not involve the male and female gametes. Of course, we shall talk about how does the pollination takes place in a plant and how does the fruit is formed. But for now, we are going to see in plants both sexual and asexual reproduction takes place. So vegetative propagation is a type of asexual reproduction in plants. As I told you just now, it's of two types: natural vegetative propagation and artificial vegetative propagation. Vegetative reproduction is any form of asexual reproduction occurring in plants in which a new plant grows from a fragment of the parent plant or a specialized reproductive structure. Many plants naturally reproduce this way, but it can also be induced artificially. For example, tuber of potato, rhizome of ginger, and there are many others that we are going to discuss now. Let us see how the vegetative reproduction takes place with the help of leaves. How are the leaves involved in the reproduction? For example, in bryophyllum, small plants grow at the edge of the leaves. You can see in this slide. Can you see the new plants growing at the edge of the leaves? It's bryophyllum. Now here, as I just now told you, in leaves, bryophyllum leaves, the new plants are grown at the edges of the leaf. So bryophyllum is the example where leaves are involved in propagation, in reproduction. So new plants are growing from the edges of the leaves. Then how are the stems responsible for reproduction? Yes, we also have stems responsible for the reproduction. Aerial weak stems like runners and stolons, when they touch the ground, give off adventitious roots, tuft of roots. Adventitious is tough, taproot is single root. When the connection between the parent plant is broken, the stem portion with the adventitious roots develop into an independent plant. For example, some examples of propagation by stem are from stolons, bulbs, corms, tuber, etc. Now let us see first how the bulbs are formed. Bulb example is onion. Yes children, bulbs. Now what is the portion that we eat in onion? Stem. We are not eating the leaf. Of course we can eat the leaves also and the roots are there, but the edible part of the onion is the both leaves and stem. Adventitious roots develop from the new stem and act to absorb water and nutrients. Contractile roots develop from the base of the bulb. Contractile roots function to readjust the depth of the bulb in the soil by shrinking and expanding. The contractile roots pull the bulb to the proper depth in the soil. That is why the part of the bulb is in the soil. The stem is also underground here. In all these cases, the stems will be underground, that I am going to discuss now. For example, tubers. Tubers example is potatoes. Now what happens here? The tubers are enlarged structures in some plant species such as storage organs for nutrients. So here the stems are the storage organs in bulbs, onion, and in tubers, potato. They are used for the plant's perennation to provide energy and nutrients for growth during the next growing season and as a means of asexual reproduction. Stem tubers form thickened rhizomes or stolons also. Now you can see the example of rhizome and stolon. Stolon is here. Example is the strawberry plant. A stolon is a stem that grows along the ground surface and adventitious roots are produced at the nodes, and buds on the runner can develop into upright shoot, and separates the plant can be formed, and separate plants can be. As you can see, they from the nodes the roots are developed, adventitious roots, tuft of roots, and from the buds the stem is developed, and they get detached from the parent plant and lives as an individual plant. This is an example strawberry plant for stolon. Then what are corms? Corm is a swollen vertical stem, solid fleshy with a papery covering. Colocasia, you know colocasia? You call it in Urdu arvi, in Hindi arvi. That is an example of a corm. Then let us see the rhizome. Rhizome are also called creeping rootstock, horizontal underground plant stem capable of producing the shoot and the root system of a new plant. Notably, the rhizomes of some species including ginger, turmeric and lotus are edible and valued for their culinary applications. Yes, you know ginger, it's very important, it adds taste to our food. So this is an example of rhizome where you can observe here the how the leaves are developed, how the new buds are formed in rhizome. Now coming to the, so far we have seen the natural vegetative propagation. Now I am going to talk to you about the artificial propagation. The first, in artificial propagation we shall do cutting, layering, grafting and also spore formation. Now first is the cutting. Cutting a plant. How does the cutting in a plant done to carry out the artificial propagation? A plant cutting is a piece of plant that is used in horticulture for vegetative asexual propagation. A piece of the stem or a root of the source plant is placed in a suitable medium such as moist soil. Some plants can be grown from leaf pieces called leaf cuttings, which produce both stem and roots. Now the example of the cutting are rose and hibiscus. This is very simple. Here what we do is very simple, children. Here we take a parent plant and we cut the part of the parent plant. It can be root or it can be stem or it can be leaf, and we put it in the moist soil and allow it to grow, so that if it is a leaf it will develop the stem and roots. If it is a stem, it will develop the roots. If it is a root that you have cut, then it will develop the stem and leaves. So whatever you have chosen, accordingly it will develop the missing parts and it will develop into a new individual. So the example of the cutting are rose and hibiscus. Okay children, you have to be careful when you are cutting with the blade. Now the second is the layering. Layering is again very interesting, as we have seen in stolon. How in strawberry plants the roots are developed at the nodes and the shoots stem is developed at the buds and it propagates. Now the development of roots on a stem while the stem is still attached to the parent plant is called layering. Yes. Now you observed in this nerium and jasmine. The examples are nerium and jasmine. Here the development of roots on a stem while the stem is attached to the parent. The stem is attached to the parent, but it is also in touch with the ground. So it will develop the new stems while it is still attached to the parent, the stem will develop the new roots. And this is called layering. A layer is the rooted stem following detachment, removal from the parent plant. Some plants propagate naturally by layering, but sometimes plant propagators assist the process. As you can see in this slide, how the roots are getting developed, adventitious roots to the stem when it is still attached to the parent. So this is an example of layering. Now let us see what is grafting. Grafting is very interesting. Now here we actually select two plants. One is seated in the soil, which is present in the soil, and another it is we cut it from the other plant and bring close to it. The one which is in the soil is called stalk, as you can see in the slide, and the other which is cut from the another plant and brought is called as the scion. We put them together, we graft them, unite them, and we wrapped around it a piece of cloth and twine around it with a string, so that both of them develop the tissues that are integrated into each other and they will form a single plant. So this is used when you want to have the selected characters. When you are selecting two characters, one character in one plant and another character in another plant, another trait in another plant, and the two traits can be united and you can get it in a single plant. So when you want to have a desired characters, you can select the grafting. I will tell you the example: mango, citrus, apple, rose, all these are the examples of the grafting. So in grafting, it is a technique where the vegetatively two stems are joined together into one. Instead of cross-pollination, two plants and producing a hybrid seed. Grafted plants use the roots and the bottom portion of the one plant. It is called as the stalk. Stalk is the part of the plant which is attached in the soil, and that is attached to the tender shoot, scion. Scion is a new plant that you are getting from another place, from the top portion of the another plant. Examples, as I told you. Mostly this question is asked in the exam. Write the difference between the layering and the grafting. In grafting, we have stalk and scion, whereas in layering we don't have. We have only single parent plant. Then what is spore formation? Spore formation is a method in asexual reproduction. It generally happens in non-flowering plants such as fungi and bacteria. In this, the parent plant generates thousands of spores which individually grows in a plant. These spores are microscopic and round in shape and grow in suitable condition. Example, Rhizopus. It's an example of fungi. Mostly you observe on the bread molds. They are the example of spore formations. They spread. Yes, they reproduce by the formation of spores. Now here children, we have one small activity here where I would like to tell you how to do it. Actually, we should take the bread. This activity is there in the textbook. You can refer it also, but still I would like to explain here. Leave the bread in open for about an hour so that it is exposed to the contaminants in the air. Place the bread in a plastic bag, sprinkle water over it to have dampness, then seal the bag, leaving some air inside. Place the bag in a dark, warm place. A kitchen cupboard close to the stove may be one option. Or you can place it next to a window with a bowl or a lid covering it from the light. Now mold will grow best in a moist environment. Mold would start growing in 2 to 3 days, but it will take a week or more to form the spores depending upon the weather conditions. Check the piece of bread every few days and add some water if it is drying. Avoid opening the plastic bag as much as you can. If you touch the bread, be sure to thoroughly wash your hands afterwards. When sufficient mold has formed, you can prepare a slide and examine it under the microscope. You will observe thread-like structures with the round structures at the top. So I will tell you about that. So before going to prepare the slide, you should know how to prepare a slide. To observe, what is our aim? Our aim is to observe the rhizopus or common mold under the microscope. Materials required are mold sample of bread, plain glass slide, cover slip, water, disposable gloves. Let's see the procedure. Place a drop of water in the center of a slide. Using a toothpick, scrape very little of the mold and place it on the drop of water. Take the cover slip and set it at an angle to the slide so that one edge of it touches the water drop. Then carefully lower it over the drop so that the cover slip covers the specimen without trapping any air bubbles. You have to be careful, no air bubbles should come. Use the corner of the tissue paper or blotting paper to blot up any excess water at the edges of the cover slip. Now view the slide with the compound under the compound microscope. First observe under low power and then under the high power. What will you observe? You will observe the common bread mold consists of fine thread-like projections called as hyphae and a thin stem having a knob-like structures called sporangia. Those knobs are called as sporangia where the spores are present. Each sporangia contains hundreds of minute spores. When the sporangium bursts, the tiny spores are dispersed in air and they again develop into the new molds, into the new structures having the hyphae and the sporangia. See children, the spores are present inside the sporangium and the thread-like structures are called as hyphae. And when the sporangium bursts, the spores are dispersed. They are spread all over and they continue their life cycle. Now the last part is the sporophyll. What is a sporophyll? A sporophyll is a leaf that bears sporangia. Both microphylls and megaphylls can be the sporophylls. In the heterosporous plants, sporophylls, whether they are microphylls or megaphylls, bear either megasporangia, thus they are called as megasporophyll, or microsporangia, as they are called as the microsporophyll. I want to show you in the slide how they are present. The sporophyll is a leaf containing the microsporophylls or the megasporophylls. That is, again, it contains the spores inside. So today we have seen what is vegetative propagation, how it is carried out naturally by the bulb, tuber, stolon, corm, rhizome with examples, and we have also seen artificial vegetative propagation, that is cutting, layering, grafting, spore formation. And we have also seen the lab activity to observe the rhizopus on bread, and lastly we have seen the what are sporophylls. So today's homework is write the differences between layering and grafting. You will get this answer in the PDF, and also write the materials required and procedure followed by you to observe the rhizopus on bread in the lab. Draw the diagram also. I hope you enjoyed watching this video and understood the concept. Thank you very much for watching this video.